everybody. Oh my gosh, it's so good to be here. This is my 5th year at the Float Conference. And I am so excited to talk to you about the research that I've been doing over the past 6 years. And I've called it the immune system blues, or how I came to terms with the limitations of experimental clinical design and got a PhD. I was thinking like Dr. Strangelove when I wrote this title, but then someone online said it was kind of like a Sufjan Stevens song, and I really liked that analogy. So like I said, it has been 5 years. 5 years since I introduced Clara. 5 years since we met her counterpart from cave woman times. 4 years since we learned about the songs that our hearts sing. 3 years, wait, did I get those numbers right? 2 years, who knows what time is. 2 years since something happened that probably no one ever heard of that involved a virus. And then last year, we became good friends with old friends in my talk about the immune system and reactivity. But this year was going to be my crowning achievement. It was going to be the crown jewel on my antibody, the pièce de résistance of 6 years of research looking at the effects of the immune system, or the effects of flotation on the immune system. But, as with a lot of things in science, sometimes our expectations don't go exactly the way that we anticipated. Or sometimes we find things that are surprising or more nuanced or harder to understand. And so today, I want to communicate how we understand that nuance and how we pick apart what we can understand based off of science that can sometimes get a little messy. And to do that, I want to introduce a concept that I'm sure all of you have heard of: the experiment. And the experiment is at the heart of what it means to do research science. When we have a question, we use experiments to come to answers. They help us to find conclusions. But understanding how to design an experiment, how to ask the right questions, how to combine questions, and then what the answers to those questions means is actually a lot more complicated than just building one. And so, how do you build an experiment? I want to start here. You know, as scientists, we look for patterns, right? We have our magnifying glass out. We're constantly looking to see how things connect, how they come together. And to go along this journey, I want to use a story that most of you, I'm sure, are very familiar with. And that is of our patron saint of floating, Dr. Justin Feinstein, and his experience with a float tank almost a decade ago. Now, as a scientist, Justin experienced floating and he noticed patterns. He realized that when he was in the tank, he became less anxious. He was able to meditate. It was relaxing. It was an experience unlike one he had ever had before. And he asked the question, can this help clinical anxiety? Now, to go from that question to everything that you've seen in the past several years took quite a few steps, because one of the issues with that is that when Justin asked that question, we didn't really know a whole lot about clinical anxiety and floating. There was some hints at previous support, but a lot of it was based off of stress. A lot of it was based off of self-report anxiety. There wasn't always data about whether people were diagnosed with anxiety conditions. And we had one that looked at generalized anxiety disorder, but it didn't look at a lot of other anxiety disorders. And so Dr. Feinstein realized that in order to adequately form a question and a hypothesis that could then be turned into an experiment, he would need to gather more data first. So before he even got to the stage of a full experiment, he created a study that would allow him to gather more information about flotation and clinical anxiety. He invited 50 people to be involved. They all had clinical anxiety of a variety of different diagnoses. They all experienced one float, and then we measured changes in state anxiety. And as this has been presented in previous years, we saw a dramatic reduction in state anxiety, and this was published in 2018. So that's great. So now we have enough information, right? So now we know that floating can reduce anxiety in people with clinical anxiety of a variety of different diagnoses, and it can do it to a pretty large level. So now that information gives us enough to make a hypothesis. And so from there we make our hypothesis. And our hypothesis is a single float session of flotation rest will lead to a significant reduction in state anxiety in individuals with clinical elevation in anxiety. Now again, I know most of you know this already, but I'm doing this because I want to talk about this structured approach and what it really means with all of the other questions we have. And so we have this hypothesis, a study was designed, this study has been taught, and a study or an experiment needs several things. An experiment needs a manipulation, so in this case it was putting people into a float tank. An experiment needs a control group, have a group of people that don't experience floating so that we can compare the two, and an experiment needs random assignment. People randomly assigned to conditions to make sure that we're getting an accurate distribution of different people in each condition. And so an experiment was designed, which has again been reported on in the past. In this study, it was what we call a randomized crossover design, in which condition 1, people were randomized to either float or watch a nature documentary, and then condition 2, they swapped, they did the opposite condition, there was a 1-week washout period in between. And this experiment was a success. We have some great data. This was presented in 2018, I believe, by Dr. Feinstein. And it shows that floating in this experiment increases serenity, increases relaxation, decreases muscle tension, decreases state anxiety, which is great. So we now have an experiment that was designed based off of previous information to test a single hypothesis. But, oh, there you go, check mark. But what if we have more questions? And see, this is key, because in the float community, we have so many questions. Whenever I talk to any of you, I am getting 1,000 different questions about what we can learn and know and understand about floating. And they're wonderful. We have so many different areas that we want to explore. But in doing so, through the scientific method and through using experiments, there's things that we have to keep in mind. Because an experiment is designed to test one hypothesis, one question. Maybe another one that's very closely related. But when you have so many questions, what do we do? And this is especially important because experiments are expensive. They take a lot of money, they take a lot of resources. We can't just design an experiment for any question or every question that gets asked. I would love to if we had infinite resources, but we don't. So what do we do? And here's the thing, is that we can't just tack all of our questions onto a single experiment, right? That might seem like the answer to our questions. Just keep asking the same question from the same data that you just collected, or keep asking new questions from the same data you just collected. There's some problems with this that are not immediately apparent, and they're called the trouble with multiple comparisons. And I want to kind of give a bit of a thought experiment to identify and illustrate why this can be a problem. So here we have a population of claras. For the purpose of our thought experiment, these are all the claras that exist. And in this population, claras either wear blue shirts or blue dresses or orange dresses. And our question is, how many claras wear blue dresses? Or what proportion of claras wear blue dresses? Now, as scientists, we rarely, if ever, have an opportunity to survey an entire population. So it's impossible to count every single person in the human race and ask them the same question. So what we have to do is we have to pick a sample. And so in this case, to answer our question, we pick a sample of 10 Claras at random. In this small sample of Claras, we have 5 Claras that are wearing blue dresses and 5 Claras that are wearing orange dresses. In this case, this sample size was adequate to answer our question because 50% of our sample is wearing blue dresses and about 47% of the population at whole is wearing blue dresses. So this little kind of experiment worked. We picked a sample. It's not really an experiment, but for purposes of explanation, we pick a sample and we ask our question. But what happens when we start asking more questions from the same setup? For example, oops, that's a sample. There we go. How many Claras are wearing sunglasses? Now, the number of Claras that are wearing sunglasses is a different proportion than the number of Claras that are wearing blue dresses. And now, the sample that we took, while, 70% of those Claras are wearing sunglasses, the population is actually 40%. So suddenly, the sample that we had is misleading. Or, to make it even more complex, what if we did it based on hair color? And what if, in this case, our question was, how many Claras are wearing pink hair, or have pink hair? Now, none of the 10 Claras in our sample have pink hair. And one of the reasons for that is because pink hair is super rare in this population. There's only 3 Claras that have pink hair. But by using the sample that we chose for our initial question, which was good for our initial question, it's now misleading us. And we're getting the answer that there are no Claras with pink hair when that's not true. Or we're getting the idea that 40% of Claras have black hair when it's actually more something like 15 in the population. Now, one of the things about this is that the more questions you ask from the same data, from the same experiment, it increases the probability of a false measurement. So the more questions you ask from the same data set, the more likely you're going to get stuff that's just not true. So we generally use the adage, one question, one hypothesis, one experiment. But this brings us back to an issue. In the same way that in order for Justin to get enough data so that he could form his hypothesis and then get the justification to make an experiment, he needed to have more information. And so we're stuck with these expensive experiments, but we still need data on which to base hypotheses so that we can build the next generation of experiments. And so in order to do that, we piggyback. Side note, these guys have an entire backstory now. I got really into drawing them one night. It's Tony the test tube. He's Italian. He loves his mother, and he's in construction. And then there's Earl the Erlenmeyer flask, and they have adventures. Like, I seriously think I need to do a side thing about these guys. But anyway, so Earl here, our flask, that's our primary hypothesis. Like, that is what we have the most confidence in. We have designed our experiment to answer this question, and so that is what we can draw from it. But we piggyback Tony on top, and we call those results, or those questions, exploratory. Because of all the problems that I just outlined, there's a high potential that whatever answers we get from them might not be true, or they might not be the whole truth, or they might just not be adequate. But they still might actually give us some insights that'll allow us to build experiments in the future. So, get to the immune system already, right? I've gone through half my talk. So, for the past 6 years, I have been studying the immune system, which is probably one of the most complex biological systems in existence. second only to the brain, which I also studied, lucky me, in grad school. You know, we had some hints that floating might affect the immune system. This was from a slide that I presented last year. Just to briefly go through it, we have reduced sympathetic nervous system activity, which is sympathetic nervous system is inflammatory, so reduces anti-inflammatory. Enhanced parasympathetic nervous system activity is the inverse. We have some papers where we recognize reduced inflammatory symptoms in individuals who have inflammatory disorders. And we also see some altered cellular immunity in a study that wasn't really well done. So we had some ideas that something might be going on with the immune system in floating. But we did not have enough data to form a hypothesis on which to build a unique experiment. We needed more data. And so we piggybacked. So this was the study that I showed you before. This is the one that's been presented on. Condition 1, Condition 2, this is where they floated or watched a nature documentary, and then they switched randomized crossover design. But on top of that, we took blood samples. And we took blood samples before and after each condition. And I've presented on this data before, but I wanted to bring it back as a reminder. Because what we found was that in response to a single float, there wasn't much difference. In fact, there wasn't any measurable difference in any of the immune biomarkers that we measured. These plots are all kind of looking at those distributions. And as you can see, all of the boxes are basically in the same place. These are box plots, so you're showing the median and the interquartile range. But basically, all you need to know is that there's just no movement. And that was a little disappointing. But I did another piggyback. So I piggybacked on top of the piggyback. So this was already exploratory. Then we did a tertiary analysis. And that was what I presented last summer. And this was really interesting. In it, what we find is that in individuals who are very high in anxiety sensitivity at baseline before they start the experiment, they tend to decrease in a particular inflammatory marker known as IL-1 alpha in the float condition, which is this line right here. So beforehand, they're a little higher, and after, they're a little lower. Again, in terms of confidence, we've piggybacked on top of a piggyback, so we're like kind of really low there. But it was interesting enough to say, hey, maybe this could inform the next study that we design. And that's what I want to talk to you about today. This time around, I wanted to stack the deck. So I already knew that a single float on its own doesn't really budge any of the immune markers that I measured. So I thought, how can I change the study design in order to kind of boost the signal that I might be seeing? And we did that in a couple ways. One was, instead of just looking at baseline immune activity, we looked at something called immunoreactivity. So your immune system, when it gets exposed to a pathogen, it kind of ramps up into action. It's reactive. And the nature of that reactivity can change from person to person, from state to state, for a lot of different things. And there have been other studies that have shown changes in immunoreactivity in response to things like yoga or meditation. And so I thought that maybe that could be a place that we could look. In order to do that, I made use of this really cool technology called the True Culture Tube. The True Culture Tube is a tube that you can draw blood directly into from a person. It has culture media inside of it, and in that culture media is an bacterial endotoxin called lipopolysaccharide, or LPS. And so once the blood hits the culture medium with the LPS, and then we incubate it for 24 hours, the blood cells, the immune cells that are within that blood, react. They start to release cytokines, immune markers, everything in between, to show that they're ready for action. And then in addition to a tube that had LPS in it, we also had a control tube that just had the culture medium and didn't have the LPS, and that was for comparison. So in terms of inflammatory markers, we also decided to try and stack the deck. We swapped out a couple of them. Those were the ones that we used in the first study. We went back, I kind of went back to the literature, I went back to my work on my dissertation. This is one of the figures from my dissertation that outlines the nature of the relationship between immune cells coming from bone marrow and how they get to the brain. And I took a couple of the metrics from here, and we replaced them kind of in this whole grouping. And I don't have time to talk about them all specifically, but I made a little animation that kind of shows how this is a mix of anti-inflammatory and pro-inflammatory markers. It's a mix of different places where they're coming from, and they all kind of have different roles. So I tried to create a wide spread of the different types of immune markers that we would be measuring in this study, and also kind of stack the deck to try and get ones that would be more involved in neuroinflammation, because those would be more related to mental health, which was where we kind of saw that result from the last study. Then I had to find a study to piggyback off of. And McKenna so nicely described it. So this was the R34, the very first NIMH-funded study that floating has ever had. And I'm not going to go into the study design because I think McKenna did an amazing job talking about it. But the really cool thing about this study is that it has 6 floats, which was another way that we could stack the deck in our favor, right? Because before, I was only looking at the impact of a single float on the immune system. And now, we could increase the dosage. We've increased the dose, more floats. We've looked at reactivity to try and find something that had more of a chance of showing an impact. We swapped out some of the metrics that we're looking at. We also added some other blood metrics to take a look. So I did everything I could to try to maximize us being able to find a result in a study that wasn't really designed for this purpose, right? Because as McKenna said, this is a feasibility, tolerability, and safety study. It was designed so that we could answer that question of, is floating feasible, tolerable, and safe? It wasn't designed to understand the intricacies and complexities of the immune system. So in this study, we took blood before and after all 6 floats or 6 chair rests. But the other thing that was hard here is that the money to analyze this data was coming directly from LIBOR, in that it wasn't coming from the grant. Because this is a piggyback study, we were getting, internal funds. And so in order to check to see if it was worth it to spend more money, we analyzed half of the data. So we looked at about half of the 75 participants that we had blood results for. And our idea was to say, let's see if there's a trend here, knowing our limitations. If there seems to be a trend, we'll analyze the rest. If not, we'll see what we can make of it. And this is what we found. So this is just an example of one of the plots. What we're looking at here is a cytokine called IL-6. If you've seen any of my other talks, I've talked about IL-6 before. It is a pro-inflammatory marker. It's part of the acute phase response. It's very early on in an infection, and it does a lot of other stuff in addition. On this side, we're looking at the results in the unstimulated true culture tube. So that was blood that went into that true culture tube that didn't have that bacterial endotoxin in it. And this is in that LPS-stimulated tube. And this is also on a log scale, so the difference between these is huge. The changes in IL-6 concentration in response to immune challenge is pretty phenomenal. The light blue is pre, the dark blue is post, and then this column is our control, and then this is the pooled flotation conditions, which again I put together the preferred and the regular pool groups for this analysis. And it's the same over here. We've got our control and we've got our pooled flotation conditions. So again, kind of looking between these, in the unstimulated condition, which is similar to what we did before, there's no difference in IL-6 concentrations from before or after either the control or the float. And it's the same in the stimulated conditions. At least for IL-6, floating didn't really change concentrations of cytokines either in a base state or in a stimulated state. And then these are all of the markers that we looked at, and we see a very similar pattern across every single cytokine. Again, this matches what we saw with respects to looking at a single float with a couple different metrics. But IL-1 beta, TNF, IFN gamma, IL-8, IL-6, IL-12, IL-10, IL-1 RA, CCL4, CCL2. These were the new additions, which interestingly didn't show any response to stimulation. So, but when I ran all the models and did all the statistics and looked at each of these, there was no difference. And so from this, in these cytokines at least, there's no changes in either resting state immune system or immunoreactivity in response to a single float as compared to a chair control condition. And then to kind of do a sanity check, we also looked at some other markers in the blood. We looked at, these are not stimulated, by the way. These are 3 different time points. It's pre, post, and 6-week is the dark blue. We looked at high sensitivity CRP. C-reactive protein is another immune marker. And then we looked at several different metrics that are usually used in like when you get a complete blood cell count done at the doctor's. I looked at hemoglobin, hematocrit, red blood cell count, platelet count, and white blood cell count. And what's really interesting about this is that it confirms that we're looking at a medically healthy sample. So we were working with folks with anxiety, but what we're looking at here, because as you can see, all of these metrics are between the dotted red lines, is that we're working with people who are basically medically healthy. And that is a really large caveat in terms of what we can say, or what the future might hold and what might come. So then, I was, that was basically our exploratory question, and then I said, well, what about all these, can I keep going? And I kept asking questions and kept doing analyses. And first I tried to recreate the effect that I had seen from the previous study that I had done. So I looked at the moderating effect of baseline anxiety sensitivity, and there was nothing. And so then I looked at the moderating effect of changes in anxiety sensitivity, or changes in anxiety impairment, or changes in depression symptoms. And again, there was nothing. And so then I flipped it and I modeled all of the behavioral metrics and looked at the impact of baseline blood metrics to see if they moderated changes in behavioral metrics. And there was nothing. And at this point, it's just kind of piggybacking on top of piggybacking on top of piggybacking on top of piggybacking. And I did so many analyses that by the end, I don't even know what I was looking at anymore. So what does this mean? Like, what are the takeaways from this data? Does flotation rest affect the immune system? That's our question. But remember, we never actually designed an experiment to study it. All of this was in the data exploration and accumulation phase. And from that, flotation rest does not have a large measurable impact on the immune system of medically healthy people, either in response to a single float or 6 closely spaced floats. But we do have some caveats there, as I've been trying to talk about throughout this whole talk. One, none of these studies were designed, none of these experiments were designed to study the immune system. We didn't have adequate sample sizes. We didn't necessarily do things that would have kind of boosted our ability to see a signal. And in fact, because of that, the only things that we would have been able to catch would have been very large effects. So it's very possible that there's smaller effects or things that we missed because we were looking through the wrong lens using experiments that weren't designed to ask the questions that we had. Additionally, we only used a subset of immune markers. I did a lot of work to make sure that they covered the gambit of all of the different types of aspects of the immune system. But the immune system is huge and incredibly complex. As I've said, there are many things that we didn't ask. But the question is, where do we go next? How do we take this information and continue our exploration down a path that might lead us to somewhere more fruitful? What I would pay attention to here is this category. We have data that shows that individuals with inflammatory disorders have symptom relief in response to floating. Is that immune-related? I don't know. But it is a really interesting place to go next. And it's one that might prove more fruitful because we'd be dealing with a population that already had an immune system that was out of balance. And so there'd be more possibility and potential for floating to bring it into balance. Whereas all of the work that we've done so far has been in medically healthy individuals. But in order to do this, we need more time, we need more money, and we need more people. And the thing to remember, especially about floating in the immune system, is that we are still in the discovery and development phase. Now, what do I mean by that? In terms of bringing a therapy to FDA approval, there's several different phases. And it starts with the discovery and development phase. And this is 3 to 6+ years, or decades. Once you have enough information to be able to get to a safety and tolerability study about your question, you can start to enter into the clinical trial phase. Phase 1, 20 to 100 people. Phase 2, 100 to 500 people. Phase 3, 1,000 to 5,000. In a process that, if it goes smoothly, takes 6 to 7 years. And then you still have about 2 years of regulatory approval. As McKenna talked about, the road that we're on is long. And we have to think about this with the end game in sight. Because we all want floating to be approved by the FDA in some capacity. But it's going to take about $40 million to get there. So I highly encourage paying attention to Dr. Feinstein when he talks about funding and identifying ways that we can make this a reality and ways that we can ask the right questions so that we can get the right information, so that we can form the most specific hypotheses and design the most elegant experiments that then help us to move forward along this path. But I'm not really worried, because this group right here asked the best questions. And I look forward to continuing to hear them and continuing to figure out how we can answer them and how we can study them and how we can move floating forward. That's me. Thank you. But wait, what's next? So I finished my PhD. I just finished my clinical internship in Tulsa, Oklahoma, where I was working at the Indian Healthcare Resource Center for the past year, working as a therapist and doing assessment with indigenous populations. And I have kind of decided that I'm going to set out on my own path. I am not looking to get into academic science or industry. I'm not interested in being a clinician. What I am really interested in is communication and creativity, and finding ways to bring science, art, and speaking together to really do something special. And in the process, I'm also looking for consulting opportunities, ways that I can support myself while I'm on that path. So if you have any ideas or you want to chat about it, let me know. If you have any ideas about any of this stuff, I am more than happy to talk. I'm super friendly, and I love a good conversation, whether it's about data or art or science or makeup or video production or whatever. But just stop me, and we can chat. Also, merch, maybe, here, coming soon? Real? I don't know. I made this slide at 2 a.m. Dylan Com and I have collaborated, and I have a line of T-shirts, and I don't know if they're available or not, but they will be maybe someday, and if so, you should buy them. Anyway, have a great day, you guys. Thank you so much.